So I'm going to be talking about a sort of separate software tool called SpringSaLaD. And I'm glad that Michael went first because some of the concepts are somewhat similar. The reason that SpringSaLaD was developed was to try to model molecular assemblies where the number of molecules involved in these complexes are too large for normal molecular dynamics calculations, especially atomistic molecular dynamics calculations. So this is, SpringSaLaD is very coarse-grained, and there have been molecular dynamics calculations that have been successfully used to look at molecular machines, which I've described here on the left. And by molecular machines, we mean that these are highly specific structures with a number of stoichiometric number of subunits, always the same number of each kind of subunit. That's what we mean by stoichiometric. The interactions between the subunits are very specific. There's a fixed size, which can be very large, like a ribosome, a discrete shape, and you can't make it unless you have a complete set of subunits. And you can use structural biology tools and even coarse-grained molecular dynamics to study these. And SpringSaLaD can be used to study them as well. And you'll be hearing a talk from Maggie Johnson tomorrow, I believe, where she's going to be speaking about a different tool. I believe she's going to talk about that, although I'm not 100% sure. That is more focused on molecular machines. But either of our tools can work on that. But what SpringSaLaD was originally designed for, and what my lab has been interested in, is something we called, way back when we first started talking about it, pleomorphic ensembles. But nowadays, that name, I should really change that on the slide, because they've been now known as biomolecular condensates. And by this, we mean very loose interactions between a variable number of components, subunit components, and therefore they're non-stoichiometric. You can have, in one condensate, you can have 100 molecules of molecule A and 20 molecules of B, and then a little bit, if you look at another part of the cell, you might find the same condensate with the same components, but there could be 80 of A and 30 of B instead of, so that's what we mean by non-stoichiometric. They're usually very weak interactions, but multiple weak interactions. So these are involved, these assemblies, these biomolecular condensates usually have multivalent interactions. And so you have combinatorial complexity similar to what Michael talked about in the case of rule-based modeling. So examples of these are signaling platforms, RNA granules, RNA droplets, lipid rafts, etc. So this is what we created SpringSaLaD for, and we kind of named it because it involves the use of springs, sites, and Langevin dynamics. So it was a nice, name, but it also kind of distinguishes between a molecular machine and a very loose combination of different numbers of components, like this SpringSaLaD, which is held together very loosely by a sticky salad dressing. So it's designed to help us reach this mesoscale between molecular dynamics and cells and maybe bridge the gap. And the talk that I'll give at noon actually shows you how we use SpringSaLaD to bridge the gap between molecular dynamics and cell-based models. So it was originally developed by a postdoc in my lab named Paul Michalski. Here's the original paper, and if you want to learn some of the physics and math details behind it, I urge you to go back to that paper. The advantages is that it deals with combinatorial complexity. It provides spatial and orientational information. The coarse-grained molecules that make this up account for excluded volumes, protein sizes, steric hindrance. So there is some similarity to doing molecular simulations, molecular dynamics type simulations. But because it's so coarse-grained, it allows you to do much bigger systems over much longer times. A molecular dynamics calculation is usually, or maybe a microsecond or two, and obviously for 1 or 2 molecules. Here you can go up to seconds and hundreds of molecules. So those are some of the advantages, but it is computationally expensive compared to Kopasi or virtual cell, but not obviously compared to molecular dynamics. So how does it work? So the point of this is not to give you a demonstration of SpringSaLaD. I urge you to, if you need it, to look at it yourselves. The idea here is to give you some idea of what it can do and how it works. And so this is sort of one of the main concepts for how it works, this idea of Langevin dynamics. In Langevin dynamics, you take an object that you want to model in terms of diffusion, and you so here's a sphere, and we give it a random force. So the force makes it take off in a particular direction, and depending on the time step, which is usually very small in the range of nanoseconds, we give it another force that's random both in amplitude and in direction. So it sort of bounces around in response to these forces, just like it would in diffusion. And the amplitude of those forces, so the direction of the forces is random. It can be in any direction, but the amplitude of those forces fits an envelope related to, that can be derived directly from the diffusion coefficient. So the average force is higher if the diffusion coefficient is higher, to make them bounce around more and faster. And the other major concept, and this is where the springs come in, is when you create a molecule from SpringSaLaD, you basically define a series of spheres to represent the shape of the molecule and how they're linked together. And the linkages are modeled in the software as very stiff springs. The actual spring constant, you almost never have to worry about that. You just use the default spring constant. The only reason for using the springs is that it transmits the force, the random force that happens on one of the spheres gets transmitted through the springs to the other sphere. So even if they aren't at that moment experiencing their own random force, they are able to respond just by being coupled through these springs to the random force that is being applied to the central sphere here. So I hope you sort of get an idea of how this works. So the total force is given by the applied random force plus the force due to the springs, and these are all vectors, so they can all move in different directions. Okay, So how do we develop molecules based on real structures? So here's just a few examples. This is part of a chromatin, I guess, and so you just represent the nucleosomes as spheres linked by, again, stiff springs. Here's hemoglobin. You can model that. You can model that in more detail if you want, but a simple way to model it would be as just 4 spheres. And here's a tubulin molecule conjugated to GFP, and you can model that as a single sphere model connected to the GFP sphere. Now, obviously, you can use more than that. Here's where just a screenshot of the actual software interface. You can see on the left here, there are different components of the model, molecules and reactions. I'll talk about reactions in a minute. And 2 types of reactions that involve internal changes as well as binding reactions. And for the molecules, we have 3 molecules that were defined in this model. One of them, nephrin, that is shown over here, that has 1 anchor sphere bound to the membrane. This line represents a membrane, and 3 additional spheres sticking out of the membrane. You can also build a model from a PDB file. So here's a PDB structure of NCK, and here's a semi-automated, you have to tell it how many spheres and where the anchor is, but this is a semi-automated way to create SpringSaLaD structures from actual molecular structures taken from the PDB. I'm not going to go through the math here, but the basic idea for reactions is that you have a physical radius of each sphere, and if you decide that that sphere wants to react or can react with another site on a different molecule, then the software automatically creates a slightly larger reaction radius. And when the 2 reaction radii overlap, because these molecules are diffusing around, and so there's some probability that they'll come together, then the probability of a reaction is derived directly from the on rate. So you need to know what the on rate is, or at least make a good guess for the on rate, if you want to run a SpringSaLaD model. So this is a very big difference from molecular dynamics, where interactions are based on potentials. Here we're translating the experimental on rate that might be measured into a probability of reaction. So let's take a quick look at an example. This is based on some work from Ani Shadaraj when he was in my lab, modeling the nephrin-Nic NWASP clustering system. So nephrin is bound to the membrane, and it has 3 phosphotyrosines. Each of those phosphotyrosines can bind to an SH2 domain on this molecule, this adapter molecule called NCK. And NCK also has 3 SH3 domains that combine to PRM domains on a molecule called NWASPs. And so these 6 PRM domains combine to any of the 3 SH3 domains, and the SH2 domain combine to any of the 3 phosphotyrosines. And so you have a situation, again, of combinatorial complexity. And all you have to do in SpringSaLaD is define which can bind with which, and then let the system take off. Here we have a simulation of a cubic reaction volume where the XY surface is defined as the membrane that holds the nephrins. There are 36 molecules and 6 nephrins, 20 nicks, and 10 NWASPs. And here's what the simulation would look like over about 100 milliseconds. So you see they all start unbound, but after 100 milliseconds, most of the molecules are bound up. Here's one that's still sort of independent, and most of the molecules are associated with the membrane. Here's a big cluster of, looks like maybe 3 nephrins and a bunch of NCKs and NWASP. And the software also allows you to do some statistics. So here's a statistic showing a histogram of the average cluster size in that simulation. And here are the kinetics as the average cluster size changes. So this histogram was taken at steady state when the system approaches a constant, and it was based on 100 runs. So you can, the software allows you to do multiple runs so you can get these kinds of statistics. This is another kind of statistic called mean occupancy, where we follow each different cluster size. And you see there's actually a probability of getting almost all, I guess this is about 36 over here. There's a probability of getting occasionally all of the molecules bound up into a single cluster. That's why this is 36. And here again is the kinetics in terms of the average of these kinds of histograms. So you can get those kinds of statistics out. And finally, my last slide, coming soon is SpringSaLaD in vCell, because as you just heard from Michael, SpringSaLaD, because you have rule-based modeling in vCell, and SpringSaLaD basically uses the same kind of idea where you have a bunch of different sites on different molecules that can bind to each other. We found that it would be fairly natural to add a new application type where we can define a SpringSaLaD model in terms of a real structure and use the SpringSaLaD simulation engine to produce the same kind of simulation results. So this is coming along very quickly due to the work of Dan Vasilescu, and this will be in vCell fairly soon. So you won't actually have to learn how to use SpringSaLaD. You'll just have to learn how to use this one application